Hi everyone, my name is Hagai Levi. I'm a PhD student in Ron Shamir and Ran Elkon's labs. In this poster, I will elaborate on one of our projects that revolves around computational methods for active model identification in biological networks. In this work, we revealed a common problem in the active model identification world of over-reporting enriched Go terms. To tackle this problem, we devised the empirical pipeline, or EMP for short, a procedure for filtering out non-specific Go terms. To measure a different algorithm based on the EMP calibration, we fashioned 5 novel AMI criteria. And finally, we presented Domino, a novel AMI algorithm that outperforms 6 leading algorithms based on this criteria. So let's start by illustrating what AMI algorithms are. AMI algorithms aim to identify interesting subgraphs in the biological network based on gene scores. Typically, after inferring the activity scores from a genomic profile, they are overlaid on the nodes of the biological network, and all the nodes of the network are rescored using some computational technique. Then, the AMI algorithm detects highly scoring modules. In order to explore the biological meaning of each module, they are commonly subjected to a functional enrichment analysis. In the beginning of this project, we examined extant popular AMI algorithms. As part of this step, we permuted original activity score sets and tested for functional enrichment. We expected to find only few very general enriched Go terms on permuted datasets, as they do not represent a real biological condition. What we actually observed is that some algorithms indeed did not get any enriched Go terms, or just few ones, while others got many. For example, here you can see that the number of Go terms J-active modules got on permuted datasets is almost as many Go terms it got on the original dataset, with great overlap. To deal with this phenomenon, we developed the empirical pipeline, or EMP for short. So first, we generate thousands of permuted datasets that yields thousands of enrichment scores for each Go term. Then, we can use these enrichment scores to generate a background distribution of enrichment scores per Go term, and use it to test how high is the true enrichment scores compared to the permuted ones. A Go term that has a significantly higher true enrichment score compared to the background is called empirically validated Go term, or EV term for short. This EMP procedure has two main caveats. First, it marks Go terms in the solution level, and not per module individually. And second, it is a computationally heavy process, and it could take even several days to fix a single solution. This motivated us to devise a novel algorithm that will have a reduced rate of false Go term calls. We call this algorithm Domino. So let's briefly review the steps of Domino. Start by dissecting the entire network into disjoint, highly connected subnetworks, or slices. Then, it detects which of these are relevant slices, that is, having a high proportion of active genes in it. Each of these relevant slices is then subjected to a refinement step, and then it repartitioned to putative modules. Finally, Domino reports as final modules only those who are enriched in active genes. We then turned to evaluate Domino against other AMI algorithms. In order to conduct a systematic benchmark in a reasonable time, we used the deep network, which is a small, high-quality global network. We also performed limited benchmark on larger networks, Fourier and String. In the main benchmark, we evaluated 6 popular AMI algorithms and Domino, so 7 in total, over 10 gene expression datasets and 10 GWASs. To compare different algorithms, we fashioned 5 novel evaluation criteria that are based on empirically validated terms. In this video, I'll focus on just 2 criteria, EHR and MEHR. So in the EHR criterion, we measure the fraction of EV terms compared to the total number of significant Go terms. In this example, the solution we got from algorithm A and algorithm B has the same EHR levels, even though they have different number of enriched Go terms. In the MEHR criterion, we calculate the fraction of enriched Go terms that are also EV terms inside each module individually. For example, here module number 2 of algorithm A has an MEHR value of 0.5. As you can see, Domino excelled in the EHR and the MEHR criteria on both gene expression and GWAS datasets. Also, as you can see, for most criteria, Domino dominates on both gene expression and GWAS datasets. To sum it up, Domino, our newly developed algorithm, outperforms the other algorithm we benchmarked. In these days, we're also developing a web-based version of Domino. So let's demonstrate what can be achieved using the Domino website. First, we need to choose an active gene file. For this demonstration, I chose a file that has 2 gene sets. Next, we need to choose a network file. Here we can choose from preloaded networks or a custom network that we have on our computer. For this demonstration, I'll choose a preloaded network. Now, let's hit execute and wait for the results. Here you can see the results of the analysis. In the left menu, you can see different solutions for different active gene sets that were uploaded. You can choose to focus on a particular module and see it in a visualized manner. Scrolling down, you can also see the genes comprising this module in tabular form and a Go term enrichment analysis conducted on this module. I'll mention here that this Go enrichment analysis does not relate to the EMP procedure presented before. Finally, you can also choose to download your analysis to your personal computer. I hope you enjoyed this demonstration. Thank you for listening. Bye for now.